Good afternoon, everyone. Thanks for sticking around up until almost the end. I'm here with Sue Washer, President and CEO of Agtc Therapeutics. All right, take us away, Sue.
Okay. So I'm just going to give just a 90-second overview of the company, and then we're going to dive right into Q&A. But I did want to kind of mention the highlights of the company. So we are an AAV company operating in the orphan ophthalmology space. And as you've heard just now on the panel, really customizing your vector for specific clinical indications is quite important. And that's something that our group has been perfecting for over 10 years now. We really believe that each vector, the capsid, the promoter, the gene cassette, the formulation, the manufacturing, the delivery, all have to be custom tailored to the specific clinical indication. And that's what we have been doing. Most important, or the most recent and big event in Agtc's life, is in July we closed on a collaboration agreement with Biogen, and wherein we're working with them on several different programs while still wholly owning a great deal of programs within the company. And that's where I'm going to kind of leave it before Q&A. This is our pipeline chart, and it does show you the programs that are partnered with Biogen versus the programs that we kept internally at the company. So X-linked retinoschisis, achromatopsia, and X-linked retinitis pigmentosa are all orphan ophthalmology indications. 2 of those are with Biogen, 2 of those we retained at Agtc, and the same is true of our research pipeline. We maintained programs internally at Agtc, and then we did partner out 3 discovery programs with Biogen. So it was a very good partnership for us, allowed us to diversify our portfolio, secure our balance sheet, and really work with another company that had an excellent level of scientific effort. Thank you.
Great. I guess first starting with the XLRS program, could you just give us an overview of maybe the disease pathology, what's the missing protein, where it's normally expressed, and just give us a basic overview.
Yeah, so XLRS is an indication that's got about 35,000 patients in the US and Europe. What's missing in this disease is there's a genetic mutation in a gene that creates a structural protein, and this structural protein holds the layers of the retina together. And the retina, while it's very thin, is very, very specialized types of cells. And if those cells can't communicate with each other, then the electrical signal that's generated by the photoreceptors can't get back through the retina and into the visual cortex to be recognized as vision. So while it's a structural protein that's missing, it dramatically affects visual functioning, in that these patients have lowered visual fields and lowered visual acuity as well.
Great. And what's the exact approach that you guys are taking to solve this?
So what we've done is that we thought in this indication what was critically important was, because the back of the eye was fragile, it's a structural protein that's missing, that a subretinal injection was not a path forward, even though a subretinal injection can get vector directly to photoreceptors, which are some of the natural cells that produce this protein. So we thought one of the most important parts of our product profile for XLRS was an intravitreal injection. And so then what we set about to do was to screen, again, in non-human primates, as you heard this panel talk about, we screened several different capsids, several different promoters, and a couple of different formulations to find the product profile that could get that protein expressed in the right parts of the primate eye from an intravitreal injection.
And what's the supporting data you guys have that really gives you confidence in your clinical approach?
So we have 2 sets of data, and for every indication that I'll talk about, we have 2 sets of data. The first set of data is in an animal model of the disease. In this case, it's a mouse model, so it's a mouse knockout model. So we have data in this mouse knockout model that we can deliver that protein, we can reform or improve the morphology and the structure in the back of the eye, and we can also increase that electrical functioning through the retina as measured by the B wave of the ERG. But in all of these ophthalmology indications, and the panel previously touched on this, lower mammal eyes are not like primate and human eyes. So human eyes and non-human primates are the only eyes in the mammal kingdom that have a fovea, a macula, an inner limiting membrane, and 3 different types of cones. So if you're going to treat primates, which is of course what we want to do, we need to test in primates, can this vector we've got deliver to the right place in a primate eye. So that's the second set of data we have. There aren't any primate models of the disease, but we can use various and sundry marker proteins to make sure that the vector is getting to the right cell types at the right levels in the right area of the eye in a primate eye. So it's those 2 sets of data that we then put together and say, okay, we can go into the clinic now.
Great. And now that you're in humans, what will you be looking for when the first set of data emerges?
Right, so this year we're expecting to report on both the XLRS program as well as the achromatopsia program. For both of these programs, they're standard dose escalation studies in patients over 18 and then an expansion cohort in kids as young as 6. And we're doing a wide range of endpoint testing as well as the safety testing. But each of these indications is slightly different. So the most important endpoints are different between XLRS and achromatopsia. So for XLRS, what we're looking at, as I mentioned earlier, as being the most important hallmarks of whether we're seeing efficacy are both visual acuity and visual fields. For achromatopsia, what we're looking at is visual acuity again and color vision, because achromatopsia is a cone dystrophy, and so it's the cones that are being most effective. And those are all approvable endpoints, and that's one of the nice things about working in ophthalmology is that there's 4 different endpoints that the FDA has guided are approvable, and then on top of that, we have the opportunity to do things like measure that electrical functioning with the ERG. We can use OCT, which is kind of like a sonogram of the back of the eye, to look at structure. So that's one of the attractiveness of working in this space is the wealth of data that we're going to be able to collect across both approvable clinical endpoints that are clinically meaningful to the patients and will show how the patients' lives are being affected, as well as these functional kind of endpoints that will look at morphology and electrical functioning.
You started to touch on this. There's some differences in pathology between the different diseases. Can you just touch a little bit about how achromatopsia is different and how you're tailoring your approach to match those differences?
Right, so achromatopsia, as I mentioned, is a cone dystrophy. So the cones are there, and we can actually visualize them through some very advanced adaptive optics out of the Medical College of Wisconsin, but they're not functioning. And the reason they're not functioning is because there's a protein missing in the membrane, the cell membrane of the cone, that needs to be there to open a channel so that the photon can enter that cone and trip the phototransduction cascade. So without that channel, the photons of light just bang up against that cone and no vision happens. So these patients are all legally blind. And so that means they have very poor visual acuity, obviously. It's less than 20/200. They also have no color vision because cones provide you your color vision. But really, most importantly to the patients, what happens is that they become very light sensitive because it's your cones that provide sight under lit conditions. So they only have rod vision, which is good. It provides you your vision when it's dark. So at all times, they're trying to keep their environment as dark as possible, so they're protecting their rods so that they at least have rod vision. Because if they don't protect the rods and the lights come on, then they'll be completely blind because their rods will wash out and shut down. So it is a very, very different kind of disease than XLRS is. And that allows me to make another comment, is that over the next 12 to 18 months, we will be in the position to have human clinical data on 4 different clinical trials. XLRS, both the achromatopsia programs, because there's 2 different genes that cause achromatopsia, and then also our program in X-linked retinitis pigmentosa. And these programs are, while they're all orphan ophthalmology, they're all pretty significant patient populations, 20, 30, 40,000 patients, they are very different methods of action, very different discrepancies in how the vision is being interfered with. And we'll have that wealth of data within Agtc to show how the technology interacts differently with these different modes of action that will allow us to make really good decisions about how to tweak those 4 products potentially, but also which of the 220 other genetic causes of blindness are going to be most amenable to bring forward into the clinic in gene therapy.
That's a really good point, and I think the last panel really focused on optimization. So, as you're looking at that wealth of data over the next 12 to 18 months, maybe just talk a little bit about the optimization efforts you guys have ongoing.
Right, so we really align very well with some of the comments made on the panel before, in that we think that historically people have concentrated very much in gene therapy on the vector. And they've kind of defined in their mind that the vector is just the capsid, just that external serotype, as people talk about. But really, a gene therapy vector, a gene therapy product, is many different components. It's the capsid, it's the promoter, it's the gene cassette, it's your formulation, it's your regulatory elements, and it's your physical delivery method. And all of those interact with each other. So we have ongoing at the company efforts to improve and optimize across all of those components, both within the company as well as our partnerships. We actually have a partnership with 4D, I think David Kern mentioned that, and a partnership with David Venables, I think David mentioned that. So obviously we'd like to do partnerships with Davids. So if there's any other David, we can work on that. But we really think that it's important to continue to push on all of these elements, and I know it adds a level of complexity to your thoughts about gene therapy, but I think it's when you understand that complexity and custom tailor it to your specific clinical indication. So the other thing we spend a lot of time on is making sure that we're working with the clinicians and the researchers that really understand these orphan indications very, very well, and that we understand not only what cells are being affected, but how those cells function in the phototransduction cascade. How does the protein function? How is it folded? How is it expressed? And so matching that clinical understanding with the specific custom tailor vector understanding, we think leads to a higher chance of success once you enter the human clinical trial.
So we've just talked about ophthalmology, but you guys have other stuff going on. Can you tell us a little bit about that?
Right, so we do have, as part of our Biogen collaboration, we just announced a non-ophthalmic indication that we're going to be working on together as a discovery program, and that is a CNS indication in ALD. We felt that as we explored the partnership with Biogen, that there was really a very natural overlap between their historic interest in the CNS space and our interest in ophthalmology. Many of the cells in the retina are actually neurons, so we thought that there was a way for us to leverage both companies' expertise and explore another field, and so it was the ALD program that we selected. And then we've historically had a program in alpha-1 antitrypsin deficiency, and we are still working on that program. As many of you may know, alpha-1 protein is the 3rd most prevalent protein in human sera, so the hurdle of the amount of protein you need to be expressing in order to have efficacy in that indication is quite high. We think we have solved those issues in a preclinical modeling, and so we'll be moving that product potentially back into the clinic.
Great, and then just the last minute, I guess just walk us through what the rest of 2016 looks like, and publicly traded companies, so how much cash do you have, and
right.
Housekeeping issues.
So as of December 31st, we had $191 million of cash on hand, and our guidance has been that cash provides us all the resources we need to get those 4 sets of data across those 4 clinical trials, and that it's at least 3 years of cash. For this year, the important milestones are the data that we'll be releasing at an appropriate scientific meeting for both XLRS and the first of the 2 achromatopsia programs. And then we'll also be filing the IND for the 2nd achromatopsia program this year. And then in early 2017, we'll be filing the IND for XLRP. So that's those 4 clinical programs that I mentioned.
Great. Thanks, Sue.
All right, thank you, Stephen.